Now, this is my fourth talk, which on world history, which will be about the subject of evolution and creation, or I might say more widely about biology. I will endeavour this time to try and keep my talk to less than 15 minutes. I think the last one went on a little bit longer than I anticipated. But as this is not my specialist subject, that's probably quite easy to do. In this subject, obviously, there are some Christians who believe that evolution is compatible with Christianity, and as I mentioned earlier, believe it's compatible to believe in hundreds of millions of years. And I'm just going to propose the evidence that might suggest that, in fact, evolution is not a valid theory, and in fact, belief in a recent creation is evidentially very plausible. Now, to do that, one has to kind of think about the original concept of evolution, which was, in a sense, a response to Paley's argument from design. Paley was a early 19th century writer, and he gave the classic example of the watch being found in the desert. If I found a watch in the desert, it would be quite reasonable to assume from all the design features of the watch that there was a watchmaker, and that this didn't just come about by chance. And I've often, in a sense, joked, well, if I found a watch in the garden on the lawn, I might be impressed by the watch and initially ignore the grass underneath it. But had I had a microscope to hand and studied the intricate details of grass, its cellular structure, and even the structure within the cells, I may have concluded the watch was but a poor design compared to the grass itself. And really that is the argument, basically, that biology points to a creator because the sheer complexity of the design is amazing. Now, when Darwin proposed his theory in 1859 in The Origin of Species, he proposed that gradual mutations over long periods of time would, or he called it changes in those days, not mutations, would be inherited and passed on, and that would create one species transforming into another. That mechanism is probably implausible, as we shall see, but the sheer design that we see today vastly exceeds the design that was apparent to the Victorians, because the more we have looked into the features, particularly of the cell, the more struck we are that there's a universe of complexity within the cell. And since then, there's been a universe of complexity within the DNA and the genetic code. All these things led quite a famous philosopher, Antony Flew, to change his mind. And I studied Antony Flew when I was at university, and he was classically the philosopher who argued against the argument from design, which is what we're talking about. But when he was confronted over the following 30 or 40 years with the accumulating evidence of complexity within the cell, he came to the conclusion, this is now a powerful argument for design. And I would agree with Andy Flew there. I think it's really so powerful that it is hard to see how one can object to that. Give an example. The genetic code was originally thought to be mainly what we call junk DNA. Sort of over 90% was non-functioning. That was until they did the Human Genome Project at the turn of the 21st century. And they studied the genetic code in great detail. And the more they have studied it, the more they realize not only is all the genetic code working on something, but it seems to be working in almost 3 dimensions, and a bit like having a sort of crossword that functions not just in the obvious ways, but functions actually in depth. So you have word sequences underneath and above the original crossword, and they're all functioning together. So it is breathtaking how complex we are talking about. And I think it suggests that we are dealing with an amazing creator. Now, probably the most recent person to kind of articulate this in a very clever way was Michael Behe in his book, Darwin's Black Box. And Michael Behe coins the phrase irreducible complexity. And he uses the mousetrap as an example. So in the case of the mousetrap, you need all the parts together for the mousetrap to work. If you don't have all the parts together, the mousetrap simply won't work. And it's like that with the cell. There are complex machines in the cell doing all sorts of amazing jobs, but unless all the pieces are together in the same place at the same time, nothing will work. And you can see this on the grand scale as well. Think about your digestion system. You need a mouth, you need an esophagus, you need a stomach, you need kidneys and so forth and such like. All these things have to be together at the same time, working in the same way. And you talk to anybody who's not well in any of those areas, and they'll tell you it has to work. So the concept of irreducible complexity shouts for design. Given the high level of design that we see, there's another interesting fact, and this comes from the fact that that design is decaying. And probably the best exponent of that is John Sanford, who wrote his book Genetic Entropy in 2005. And John Sanford, who had studied genetics for 30 years, believing in evolution, changed his mind and began to look at some of the weaknesses of evolution. And one of the most obvious ones was that the level of mutations that each generation inherits increases over time and can't be got rid of. Natural selection can't get rid of them quickly enough. And so John Sanford came to the conclusion that time is not a friend of evolution, it's actually its enemy. Time will destroy a good design, it won't create a good design. In fact, there's very little of any evidence of any beneficial mutations that, in a sense, add genetic information. There are some sort of mutations, like you might have a wingless beetle on a windy island, and the loss of wings would actually be a genetic advantage to the beetle. But that is actually a loss of information. Sickle cell anemia may give you, the sickle cell in the blood might give you some sort of protection from malaria, which gives you some, you might say, protection within areas of West Africa where malaria is rife. But sickle cells are themselves a loss of genetic information, not a gain. And so losing genetic information, even though it may give you, let's say, bacteria might get some extra protection from the sort of methods we have of destroying bacteria, that loss of information is not the type of genetic change required to produce evolutionary progress. That will actually just destroy original information. And so, as I say, John Sanford put forward a very compelling case that this is what's going on, that we're seeing a loss of genetic information. And I'm just going to show you a little slide on this, which is interesting in light of what has been going on at present, if you might think, with our current COVID-19 situation. And we notice the mutations that are, as it were, bedeviling us, as it were, because the things themselves change all the time. So I hopefully will be able to pull up now the PowerPoint. And so John Sanford made this observation that we see genetic entropy in action when it comes to certain particular diseases, and the one he's looking at is this particular disease that is associated with the Spanish flu. And the observation is that the disease itself becomes more and more, or the virus itself becomes more and more dysfunctional as time goes by because of the level of mutations that's going on. And viruses are interesting because the level of mutations in a virus are very high. And so these mutations aren't adding information, aren't making the virus in many ways stronger. Actually, they're making it weaker. And so the idea that life could be around for millions of years makes no sense, because millions of years of mutations in the genetic code would actually make all of us genetically unviable. And so that's a very powerful tool for understanding that life is not only designed and created, but it must be quite recent. Now, another person who's looked at this information is Nathaniel Jensen, and he's been looking at the level of mutations in mitochondrial DNA. Now, mitochondrial DNA is useful to study because only the female keeps that DNA, and therefore it can be studied in isolation from the fact that most DNA is the product of male and female inputs. And so mitochondrial DNA allows you to study the level of mutations that you see. If the creatures concerned had been around for the ages designed or designated by evolution, one would have expected, in the case of humans, a significant number of mutations in the mitochondrial DNA. And this is that red chart here. And what we tend to find is that we don't see that level of mutations. We see something that is much more consistent with 6,000 years. And that's what we're finding. And this same result was found in looking at the mitochondrial DNA mutations for fruit flies. What we expected to see in evolutionary terms is this red chart, if it's been around for the millions of years that they had said. But what you actually see is very few mutations, and it's much more consistent with 6,000 years. Likewise with roundworms, where we've been looking at the mitochondrial DNA. There, the mutations, what we would expect in the red bar for evolution to be true, the millions of years involved there. But what we find, in fact, is something that's consistent with thousands of years. And likewise with water fleas. And those are the four areas that this particular study has been done. But wherever we have looked with mitochondrial DNA, the level of mutations is consistent with those particular animals being around thousands of years, not millions. And I think that that's a really powerful piece of evidence. And so I trust that you'll begin to see that when we look in the realm of biology, there is definitely good evidence of believing not only is life designed, but life was recently designed. And I think that that supports the position that I've been looking at for Genesis 1 to 11. So thank you for listening.